Welcome back friends, welcome back in another video tutorial for microbiology with Shagnik. So today as promised we will going to talk about lactic acid fermentation. So in glycolysis the NAD plus which was converted into NADH will be regenerated via an enzyme called lactate dehydrogenase, and during that time the pyruvate will be converted into lactate. That is the idea of lactic acid fermentation, and here the end product is lactate. It may occur especially in muscle cell and also in some bacterial species, mainly lactobacillus. Okay, now in muscle cell we have learned that pyruvate will be converted into lactate. Okay, now this lactate, when it is required, can be transferred to the liver, where with the help of liver lactate dehydrogenase it will be converted back into pyruvate, and then via gluconeogenesis it will be converted back to the glucose. Okay, and then when it is required that glucose should enter in the glycolysis, it will be transferred back from liver to the skeletal muscle, and there glucose will be converted into pyruvate. And now if in that time lactate production is required, then lactate again will produce. Okay, so that is how you can see that there is a cycle that is going on, and this cycle is especially known as Cori cycle. Okay, from the name of its scientist Carl and Getty Cori, who have won the Nobel Prize for Medicine as well as in Physiology in the year 1947 for studies on glycogen metabolism and blood glucose regulation. So here you can see I have written the things that I have already explained you, that is in muscle cell and certain bacterial species such as lactobacillus, NADH which was generated during glycolysis is oxidized and converted into NAD plus, and during that time pyruvate is converted into lactate, and this process is called lactic acid fermentation. Lactic acid fermentation is catalyzed by the enzyme lactate dehydrogenase we have discussed, which converts pyruvate into lactate. And in animal, lactate which is formed in the muscle can be transferred to liver, where it can be recycled into glucose. Okay, and when glycolysis of this glucose is necessary, this glucose will be transferred from liver into skeletal muscle, where glycolysis will happen and it will be converted into pyruvate, and then from pyruvate via LDH again it will be converted into what? It will be converted into lactate, and that's how you can see a cycle is going on, which we will call as Cori cycle, according to the name of its discoverers Carl and Grady Cori. Okay, and that is the idea of our lactic acid fermentation. So I hope this video will be helpful for you. Thank you.